Hi, everyone. My name is Kelly Matsunaga, and I'm going to be talking about some research that my co-author slash doctoral advisor, Selena Smith, and I recently published on the morphology of palm fruits and what they reveal about the fossil record and evolutionary history of palms. So let's dive in. I'll start by talking briefly about the fossil record of palms and why we decided to study living palm fruits. I'll show you the results of some of our phylogenetic analyses of fossil palms. And finally, I'll summarize some of our sort of proof of concept work that we did to demonstrate that these relationships are probably pretty accurate. All right, so before I dive in, I just want to make sure we're all on the same page about this. So palms are currently subdivided into 5 subfamilies, 29 tribes shown here in the tips of this tree, about 181 genera and over 2,500 species. So I'll be using this tree diagram throughout the talk to illustrate several points. And this is based on the genus level phylogeny of Baker et al. 2009. All right, so the fossil record. Palms have an exceptional fossil record among angiosperm families. We find their fossils all over the world. Their tissues are fibrous, and so they preserve pretty well. And importantly, they have really distinctive morphology and anatomy, which is difficult to confuse with other lineages. So the earliest unequivocal evidence of palms in the fossil record are leaf compressions that start to show up around the Coniacean of the late Cretaceous. This is around 90 million years ago. Now, by the end of the Cretaceous, about 66 million years ago, palms are already widespread. Macrofossils are known from Cretaceous localities around the world, some of which are shown in this map here. In equatorial regions of the late Cretaceous, they also form a palynological province, shown here in green, in which palm pollen makes up over 50% of pollen grains within assemblages, indicating that not only were palms widespread by this time, but also abundant, at least in some regions. All right, so the fossil record provides pretty clear evidence that there was a major geographic range expansion during the late Cretaceous. But one of the things I'm interested in is whether this went hand in hand with the evolutionary diversification of modern lineages. So when did all of this diversity that we see in modern palms originate? Now, molecular dating analyses indicate that this Cretaceous phase primarily involved the stem lineages of the family and its subfamilies, with most tribes and genera, this portion of the tree, originating later. But what these estimates do not currently take into account are the affinities of many palm fossils from the time when palms were abundant in the fossil record. And so without knowing their phylogenetic relationships among palms, it's difficult to incorporate them into these types of analyses as calibrations or other types of data. So to get at this question about the early evolutionary history of palms, we decided to investigate the phylogenetic relationships of fossil fruits, some of which are shown here. And this is because reproductive structures often have a lot of phylogenetically informative characters. However, we quickly discovered that most of the information we needed on modern palms to do this study was not available in the literature. And so we supplemented our literature review by micro-CT scanning fruits from herbarium collections. So micro-CT is a form of high-resolution X-ray tomography that produces 3D images of a specimen from 2D X-ray images, like this one here, similar to medical CT or CAT scans. It's fast, non-destructive, and data sets can be sliced at any angle, such as this coconut data set, which we're seeing in transverse section. So here are some of the CT scans. These are fruits all seen in transverse section. And so one thing I hope you can see, at least immediately, even if you're not exactly sure what you're looking at, is that there's a lot of variation here. Some fruits have 3 seeds, others have 1. There's a lot of variation in the amount of sclerenchyma and other structures. Now, this variation is important because it allows us to distinguish major groups of palms based only on their fruits and use that information to infer the evolutionary relationships of fossils. So just to step back and take a look at the bigger picture of what we're doing, our sort of methods and workflow. So we have CT data on living palms, we have some fossils, and our end goal is phylogenetic trees with fossils as tips. So how do we get here? So I took my CT data, supplemented it with information from the literature, because there's a lot of information in the literature out there. And we use this to build a data set of fruit characters for the family. This matrix, as well as information from fossils, DNA sequence data from living palms, were all used together in phylogenetic analyses to build trees that included fossils as tips. We scanned 207 species of extant palms, incorporated 5 previously published fossil fruits. The morphological matrix contained 45 fruit and gynoecial characters. I used 10 chloroplast and nuclear genes, all from GenBank. The final data set was sampled at the genus level. And all the results I'll be showing today were from maximum likelihood analyses performed in RAxML. All right, so let's look briefly at the fruit data. So this is a amorphous space plot, which is essentially an ordination based on a principal coordinate analysis of our matrix of fruit characters. When we look at this plot divided by subfamilies, we see there's a lot of overlap between the subfamilies, with Ericoideae being an exception. It occupies a unique region of amorphous space, although part of this is in that region of overlap. When we look at the tribe level, we see that tribes within subfamilies tend to show less overlap with one another. And sort of as expected, closely related tribes tend to be closer to one another in the amorphous space or overlapping, such as Ericoideae and Euterpeae here, which is inside of it. So now for the fossils. These are all of the fossils we used. These are previously published. 2 originate from the Deccan Intertrappean beds of India and are about 66 million years old. These are Palmocarpon drypetioides and Hyphaneocarpon indicum. Friedmannia mesolensis is from the Meso oil shales of Germany, and Nypa bertini is from the London Clay Formation in England, and both are approximately 47 million years old. And finally, there's Coryphoides poulsoni, which is from the Agat Dal Formation in Greenland, and it's about 62 million years old. We did a series of analyses on these fossils. First, we performed what are called total evidence analyses, which included both molecular and morphological data with all of the fossils. However, these analyses can make it difficult to evaluate the support for placement of each fossil within a clade, because when we combine data sources, we also combine their uncertainties. So to disentangle these uncertainties and get an idea of how well supported each fossil was in its respective clade, we did a series of analyses using just the morphological data paired with a molecular backbone tree to constrain the position of extant taxa, and then analyzed each fossil individually. So let's take a look at these results. Now, everything I'm showing is based on these backbone analyses, since the results of all of our analyses were consistent. All right, so Palmocarpon drypetioides was placed in subfamily Ericoideae in tribe Coccoceae, and within the crown group of its subtribe Adelaeinae, which is shown here. Hyphaneocarpon indicum is nested in the subfamily Coryphoideae in tribe Boraceae, and in its subtribe Hyphaneae, with Bismarckia and Saturnala. This is consistent with what we found using fewer characters when we originally published on this fossil. Nypa bertini comes out in subfamily Nypoideae, sister to living Nypa fruticans. And this is not surprising given it was originally placed in genus Nypa, but we included it as a test of our data set. Coryphoides polsoni was placed in Coryphoideae, as its name implies, tribe Trachycarpii, and within the subtribe Livistoniinae. And finally, Friedmania messalensis comes out within the Ericoideae in tribe Ericae, within the Pacific clade of tribe Ericae, although support for any particular placement within that clade is weak. Now, because genus level relationships within Ericae are poorly understood, and some other reasons I don't really have time to get into here, we decided to cautiously treat Friedmania as a member of the Ericae crown group rather than a member of any particular clade within the tribe. All right, so we have 5 fossil fruits that we can place with pretty high support in several different lineages within palms. So this is very promising. But what I think is reasonable to ask is, how do we know these phylogenetic relationships are accurate? And so to test this, we performed a series of what we are referring to as phylogenetic pseudo-fossilization analyses. The idea is that we're analyzing extant taxa in the same way as the fossils to see if we can place them back into the tree accurately based only on the morphological data. So for each analysis, we removed a single extant taxon from our constraint tree. We then analyzed the morphological matrix with that backbone constraint tree we just created to see the inferred position of that taxon based on the morphological data. To evaluate the accuracy of these placements, we calculated pairwise Robinson folds, or RF distances, between the correct placement and its inferred placement. We also looked for the shallowest, well-supported node within the tree and asked, does this node reflect the true relationship of this taxon? In this case, we're looking at genera. This is important because when we're dealing with fossils, we have no way of knowing how accurate its placement is, but we do have metrics for estimating uncertainty based on the data, such as bootstrap values. So, does a high bootstrap value indicate we have correctly placed a fossil? Okay, so we performed 40 of these analyses for genera throughout the phylogeny. All the taxa chosen exhibited a range of data completeness that matches that of the fossils we analyzed. Overall, 24 out of 40 genera were placed correctly to either the tribe or subtribe level in the highest likelihood tree. And in nearly all other cases, the correct placement was encompassed by the best supported clade, indicating that high node support is a reliable indicator of accuracy. However, I said nearly all because 2 genera were not. These were Roystonia and Sclerosperma, both of which were placed in Ericae with high support, even though clearly they're not part of Ericae. So this is sort of a worst case scenario that we were hoping we would not encounter. However, after careful exploration of this, these taxa actually reveal something interesting about the data and have useful lessons for understanding phylogenetic relationships of fossils. So both taxa have several things in common. They are both the only genera within their respective tribes, and both lacked morphologically similar sister taxa in our tree. This makes more sense when looking at them in the morphospace. So here's Roystonia, here's Sclerosperma. Both taxa plot within Ericae morphospace, far away from their closest relatives. So Roystonia's closest relatives are Reinhardtia and members of Coccoceae. Sclerosperma's closest relatives are Podococcus and Arania, which are way out here. So because these genera lack morphologically similar sister taxa in this data set, removing them alters the distributions of character states in the tree, making it more difficult to place them back into the tree correctly. And in fact, this was a problem for nearly all monogeneric tribes. Most of the genera that were placed incorrectly form monogeneric tribes, and only 2 such genera were actually placed correctly. in the best scoring trees. So these were Pelagodoxa and Urania, both of which actually have morphologically similar close relatives in the data set. So here and then here. So this leads us to several takeaways. I think we can accurately place fossils to tribe and subtribes with several caveats. First, taxon sampling matters a lot. As we saw with Sclerosperma and Roystonia, incomplete taxon sampling can lead to incorrect placement of taxa. And secondly, uncertainty and node support must be considered. Now, we used bootstrap support values above 95% as our cutoff, because in some cases, we saw support values of up to 90% for incorrect placements. Now, in the case of these fossils, I think we can check all of these boxes. Our taxon sampling was extensive, and node support was high for all taxa, with any uncertainty narrowly restricted to the clades in which these fossils were placed. Okay, so going back to our main results, now many of these fossils are much older than current estimated ages for their respective groups, some by as much as 40 million years. And so this implies that Cretaceous diversification of palms involved the crown groups of subfamilies, tribes, and even subtribes, and not just the stem lineages of major groups. And so this has important bearing on how we understand the environmental context of palm evolution, because climate and environments, as we know, have undergone radical changes over the last 100 million years. As we shifted from warmer and drier environments of the Cretaceous to the wetter and more equable climates of the Eocene, and then to the cooling and drying trends in the Oligocene towards the present, we can imagine these might have had important effects on the evolutionary history of palms, 90% of which inhabit tropical rainforests today. So just to conclude, morphological data on palm fruits can be used to resolve the phylogenetic relationships of fossil palms. Our analyses show that several modern tribes and subtribes had diverged by the end of the Cretaceous or the Eocene, and this implies that the Cretaceous diversification of palms produced many of the lineages we recognize today. So with that, I would like to thank you all for your attention. In particular, the organizers of Palms 2021 for inviting me to give this talk, my funding sources, collaborators, students, and the many natural history collections that made this work possible. So if you have any questions, feel free to email me at matsunaga@ku.edu, or check out the paper, which is in the American Journal of Botany. The title is the same as the name of this talk. So thank you.